Hello everyone. It is an honor to share with you our team's perspectives and insights on the feasibility of using pathobiome strains for live biotherapeutic products. As our understanding of the role of gut microbiota in human health deepens, people are becoming aware of its close association with gastrointestinal health and diseases of other organs. In addition to traditional beneficial bacteria such as lactobacillus and bifidobacterium, non-traditional beneficial bacteria like Escherichia, bacteroides and Enterococcus also play important roles. Some scholars refer to these non-traditional beneficial bacteria as pathobiome strains, which may interact with the host and impact health. How to assess their clinical pathogenic and potential benefits? Can they be transformed into live biotherapeutic products? To address the first question, we must recognize the inherent variability among individual strains of the same species. Even with pangenomic approaches, it's challenging to accurately categorize species as good or bad. Therefore, scientific evaluation should be conducted at the strain level. Specifically, attention should be paid to the key functional genes and their regulatory mechanisms within the strains. The presence or absence of these functional genes determines the potential of the strains to impact human health. Furthermore, the influence of ecological niches must be considered, especially the interference of symbiotic microbiota, which may lead to differential gene expression patterns of strains in different ecological niches, thus exerting dual effects. Lastly, the interaction between bacteria and drugs is also a factor to consider, as the gut microbiota may affect the efficacy, toxicity, and bioavailability of drugs. The development process of LBP is similar to that of conventional drug development, but it has certain peculiarities in terms of drug safety, efficacy, and quality management. In terms of safety, the first concern is the common occurrence of bloodstream infections caused by bacterial translocation. This also includes tolerating salvage therapy antibiotics, posing risks of genetic transfer, produce toxins, and metabolized drugs. The study of LBP efficacy is complex, which is related to bacterial quantity and metabolic intensity, because live bacteria have the ability to self-replicate. The generation of metabolic products is dynamic and susceptible to the influence of ecological niche. The quality management of LBPs often overlooks the impact of dead bacterial components. Insufficient research on the efficacy and safety of these components may become obstacles in the development of LBPs. Here, we propose possible research directions and application approaches. Firstly, we need more studies on gene functionality to reveal the genetic background of strains. Simultaneously, it's essential to establish microbial community models to further explore the impacts of ecological niche. Additionally, attention should be paid to bacterial translocation and genetic transfer in clinical trials. In terms of development form, besides large-scale screening for candidate strains, using genetic editing to balance the safety and efficacy of LBP is feasible. There have been numerous scientific studies in this regard, but the road from successful scientific research of our LBP to commercial success is lengthy. Additionally, bacterial metabolites offer a potential alternative, extracting large molecule active components challenging to synthesize chemically via bacterial fermentation or inactivated bacteria such as postbiotics. In summary, it is inappropriate to simply classify non-traditional beneficial bacteria, and even pathobiome strains, as beneficial or harmful. Extensive research into the genetic functions, metabolism, and microbial interactions of each strain is necessary. A thorough understanding of the genetic background of the strains is essential for their translation into LBPs. This translation can be facilitated through various methods, including large-scale strain screening, gene editing, or extraction of bioactive metabolites. Although these studies may be lengthy and challenging, they are worth further exploration.